Good morning, good afternoon, and good evening, everyone from wherever you're joining us, and welcome to today's webinar titled The Scientific and Operational Elegance of Study Replication with Real-World EHR Data, a vitiligo and alopecia example. I'm Sydney Otamanchek from the events team at scientist.com, and I'm happy to be your host for today. This webinar, hosted by experts from Panalgo and OM1, will look at the scientific and operational details behind study replication using vitiligo and alopecia areata as an example. I'd like to thank the sponsor of today's event, Panalgo, for making today possible. And now, without any further delay, I'm going to welcome Meg to the floor to introduce the webinar and our speakers. Take it away, Meg.
Thank you, Sydney. Good morning, everyone. Good afternoon, good evening. It's lovely to be with you all today for our webinar. Thank you so much for joining us. My name is Meg Richards, and I'm an executive director at Panalgo. With me today is Mike Munsell, also from Panalgo, and Catherine Starzik from OM1. So Panalgo and OM1 have a long-standing partnership, which has been made even stronger by the recent agreement to make OM1's datasets available on demand within our Instant Health Data or IHD platform. To demonstrate the power of OM1 data in our platform, we chose two autoimmune conditions for which there is no cure, but active treatment pipelines. So in keeping with the title, we have four objectives for this webinar. The first is to understand the vitiligo and alopecia areata landscapes. The second is to appreciate the scientific elegance of study replication. Thirdly, we want to demonstrate the operational elegance of study replication using the combined power of an analytics platform and some very rich data sources. And then finally, we want to highlight the richness of a multimodal data source such as OM1's. Let's start with a little bit of background on our two autoimmune conditions. Catherine, in her section a little bit later on, is going to put more sort of color on the epidemiology of these two conditions, but I'd like to provide just some general definitions here. So vitiligo is a disease that causes areas of the skin to lose color, resulting in patches of lighter skin. Some people develop it in a few spots, others have more widespread loss. It is an autoimmune condition wherein the body attacks its own melanocytes or pigment cells. So the prevalence of vitiligo is thought to be about 0.1 to 0.3%, with the highest rate seen in older Hispanic, Latino, and Asian American patients. The global lifetime prevalence is as high as 2%. Current treatment options are varied. Catherine's going to talk a little bit more about these, but there are topical medications such as corticosteroids, tacrolimus. There are ingested medications like prednisone, phototherapy, surgery, cosmetic approaches, and depigmentation. This picture on the left is Winnie Harlow. You may have heard of her. She was a contestant on America's Next Top Model, I think in 2017 or thereabouts. Pretty successful model who really celebrates her vitiligo. There's a healthy pipeline of treatments, about a dozen products that include work that's being done by AbbVie, Pfizer, Merck, and Clinuvel. Opzelura, which is an Insight product, was approved in July of 2022, so that's on the market. One of the interesting things about vitiligo is that a retrospective study of over 3,000 vitiligans, and that's what you call them, showed that comorbid autoimmune conditions, not surprisingly, occur in approximately 23% of patients. And those include things like thyroid disease, rheumatoid arthritis, IBD, lupus, and type 1 diabetes. The psychosocial burden that comes with vitiligo is something that's very interesting and very prominent. It's understandable that, driven by the need to conceal the lesions and to meet societal beauty ideals, there's an attendant stress, anxiety, depression that can follow in the wake of these visible differences. And then just a little quick bit on alopecia. Alopecia areata is another more common autoimmune disease than vitiligo, causing sudden hair loss on the scalp, face, and sometimes other parts of the body. Alopecia means bald, and areata means patchy. Like vitiligo, it is an autoimmune disease because the body's immune system mistakes healthy hair follicles as dangerous tissues and attacks them. So, as I mentioned, the prevalence is a little bit higher. About 2% of people globally will experience AA in their lifetime. Most develop it early. It happens more often in women than in men. And studies show that prevalence is higher among non whites versus whites. People with alopecia areata have a genetic predisposition for it, but it's also important to have an environmental or stress trigger often to be a factor in the development of alopecia. I think that's true of almost all autoimmune conditions, is that you need the genetic predisposition and a sort of a stressful event to have it manifest. Treatments depend on the age, the extent of the hair loss, duration, and location, but they include, again, topical corticosteroids, minoxidil, which is Rogaine, bimatoprost for glaucoma. There are JAK inhibitors and a treatment called contact immunotherapy, as well as cosmetic approaches such as wigs, shaving, tattoos, etc. So the alopecia landscape looks a lot like the vitiligo pipeline. There are two JAK-STAT inhibitors that have been approved, Litfulo and Olumiant. There's also a similarly healthy pipeline with AbbVie, Aslan, Amgen, and others contributing developments at phase 2 and phase 3. As with vitiligo, alopecia is also associated with other autoimmune conditions, and a lot of them are the same: atopic dermatitis, rheumatoid arthritis, lupus, type 1 diabetes, celiac, and others. Not surprisingly, as with vitiligo, the psychiatric burden of moderate to severe alopecia is rather high, owing to the potential to feel rather disfigured in appearance. And here's an interesting fact. The vitiligo and alopecia cohorts overlap. Depending on what you call the underlying population, there's about a 3% overlap in the condition. This just shows you some of the levels of alopecia. You can have patchy, which is limited to certain areas. You can have totalis, which involves the total scalp, or you can have universalis, which involves the entire body. So now let's hear from Dr. Mike Munsell, who's going to talk about replication of studies in our environment. Mike will share both the scientific and operational elegance of study replication in our IHD platform. Take it away, Mike.
Thanks a lot, Meg. So as Meg mentioned, from an epidemiological perspective, the concept of reproducibility is not new. So you clearly need reproducibility to establish causality or even correlation, really. So the Bradfield-Hill criteria, which includes 9 elements for establishing a cause and effect relationship, established several decades ago and is listed up here on the screen, includes consistency or reproducibility as one of the criteria, which states that consistent findings observed by different people in different places among different samples strengthens the likelihood of an effect being a true effect, which is fairly intuitive when you think about it. Now, taking this concept, though, into the real-world evidence space, there has been a lot of discussion on the need for reproducibility in RWE analyses in order for them to be used in more critical activities such as regulatory decision making. In order for RWE analyses to be reproducible, there needs to be clear communication and documentation about things such as the study design, the analysis, and as well as the implementation of this into a final published study. ISPE and ISPOR, which are two key stakeholders of RWE, created a joint task force to build a template for documenting these types of study elements in a way that promotes reproducibility in RWE analyses. So if you haven't seen it yet, it's called the Harper Protocol, and it was published 2 years ago. And the publication includes clear templates for documenting RWE studies that help to promote reproducibility. The Harper Protocol focuses on pretty much all aspects of a study, from the study rationale to even the metadata of the underlying data source. But the majority of the document focuses on methodological design of the analysis, including clear structured definitions for variables and timeframes. An example table from the Harper publication is on the screen and gives an example of identifying a cohort in a case control study design, where time zero is the date of initiation of either treatment, and the study population inclusion criteria attempts to mimic therapy initiation in a randomized trial framework. Panalgo's Instant Health Data Platform, or IHD, which is a low-code analytics platform designed to execute real-world data analyses, is similar to the Harper protocol in the sense that IHD defines cohorts, variables, and timeframes using a structure that's reproducible and independent of the data source that's being used. Similar to generating program code in SAS or R to execute an analysis, in IHD, cohorts and outcomes are defined by identifying key encounters and events based on diagnosis and procedure codes. Relative and fixed timeframes can be created to identify treatment patterns and study outcomes. And lab results and notes can be extracted from the data source to supplement findings. However, unlike coding, the platform ensures that a standard definition for key variables can be picked up and placed on top of a different data source with reproducibility and a low probability of coding errors. So we'll take the example of the operational definition for cohort identification from the Harper protocol, which is designed to foster reproducibility, and see how these types of variables are also defined within the context of the IHD platform. So take, for example, the comparator here, where the study population is defined by the presence of an exposure to a DPP4 inhibitor. In IHD, we're able to identify patients with exposure to a DPP4 inhibitor by using what's called the presence of encounter measure, which essentially locates a claim. In this case, we're looking at administrative claims data for a specified event using data elements such as code lists and also setting type. And within this presence of encounter measure, we can then identify a specific medication event, which uses a user-defined NDC code list to search for evidence of a DPP4 exposure. So this is similar to what's listed in the Harper protocol, where you're identifying using an NDC code, and within the context of this project, that NDC code can be used for multiple different data sources. So the code list that's being referenced here will follow this project in its many iterations. For a patient, if that DPP4 medication is identified, the date the DPP4 inhibitor was prescribed can be tagged as something such as an index date, which will be useful when determining a washout period or a baseline period. We can also specify the care setting in which the DPP4 was initiated and identified, such as a clinic or emergency department event. In the Harper protocol example here, there's no specified care setting that's required. So in IHD, leaving that blank opens up any possible care setting to be identified. And finally, based on that date of first observed DPP4, we can create a time period in IHD that looks back 180 days and create a similar presence of encounter measure to ensure that no other DPP4 is observed during this time period. And within that washout period, we can also create measures for continuous enrollment and other inclusion criteria that want to be selected. So this concept of clear but flexible structure, which is recommended in the Harper protocol, the advantages of analytic platforms that are also designed in a similar fashion is the concept of reproducibility, because as you can see with this example, the structure is independent of a specific data source that's being used. The codes that are being referenced can be applicable to many data sources. So going back to the main theme of our presentation, IHD was used to publish a study last year that evaluated the burden of incident comorbidities among patients with newly diagnosed vitiligo. The original analysis made use of market scan commercial claims data, and in order to replicate the analysis on a different claims database, all we had to do was point the original project to this new data source. So all cohorts, code lists, variable definitions remain constant, and the only thing that is changing is the underlying data source that is being referenced. So I'll spend a brief moment discussing the overall study design of this published analysis, and then we'll dive deeper into the actual reproducibility of the study findings and what we saw across the databases that we used. So as I mentioned, the original analysis was conducted using the TESA conducted using market scan commercial data over in the replication, we used Panalgo's TESA closed claims database. So TESA is a closed claims US-based database that includes administrative and claims data for over 170 million patients across commercial payers, Medicare Advantage, and also Medicaid. For the purposes of this study, we are only using the commercial database for reproducibility purposes. The vitiligo cohort was diagnosed, included a 1 inpatient or 2 outpatient claim for identifying index. And these patients were matched based on age, sex, and region to controls with no evidence of vitiligo in the database, using a ratio of 4:1. So for the cases, those with vitiligo, we did require 12 months continuous enrollment prior to the first vitiligo diagnosis. And given that the analysis that was published wanted to identify incident autoimmune and also psychiatric comorbidities among an incident vitiligo population, no vitiligo treatment or diagnosis, as well as any autoimmune or psychiatric comorbidity of interest, was allowed during the baseline washout period. Patients were then followed until the first of either the end of continuous enrollment or a diagnosis of the comorbidity of interest, requiring at least 12 months of continuous enrollment for the study population. So as I previously mentioned, within the IHD platform, all a user needs to do to change the data source but maintain the study design is to switch the data source that is referenced in the project setup. So given that data sources are mapped to a common data model within IHD, when a data source is switched, any discrepancies across the databases are identified by IHD and required to be resolved by the researcher prior to rerunning the analysis on the new data set. So this actually, there were limited discrepancies in this current replication study among the vitiligo patients, given that all required variables were fairly similar across the claims data sets. They were both administrative commercial claims data sets. But I give an example here of what this tends to look like. So say a new data source had different setting locations coded for the identification of vitiligo. So maybe in the outpatient setting, there wasn't a code for an office location, but there was a clinic and also surgical center. So this is kind of the nuance of the coding of the variables within the data sets. So within the variable, that would be highlighted. and brought to the researcher's attention. And then you'd be able to go in the platform and update the specific codes that were included in the outpatient encounter. So in this example, it's including all of the codes that were available in the previous data set. And then based on the highlighted changes that you're seeing, you could select the outpatient location settings that were applicable to the new data set, which would allow you to have a measure that is not highlighted anymore and you can run. So while this is very helpful for understanding how to change the analysis based on the data set, it's also extremely helpful for documentation, particularly in the context of replication, of knowing what things needed to be adjusted in terms of variable names and other aspects across data sources if you are looking to document and replicate the analysis. So now into what we actually saw when we swapped out the data set, maintain the study design across the two data sets and looked at the replication of the findings. So what we did find was that the primary conclusion did hold. So with both data sets, they identified a higher incidence rate of any psychiatric or autoimmune comorbidity in the vitiligo cohort compared to the controls. So we saw a slightly lower incidence overall with TESA data, but this is primarily due to a smaller time window by about 3 years compared to the market scan data that was used in the analysis. And also some of the time frame spills over, not in the market scan data, but in the TESA data over the pandemic with known kind of lower hospital healthcare utilization over that time period. But overall, the magnitude and primary finding is similar comparatively between the cases and the controls. And anything outside of that, I mean, there were some expected heterogeneity across these study populations. These are inherently different patients. And so, not everything is going to look exactly the same. And in terms of replication, the primary study finding of higher incidence rates was confirmed, but there was some difference in which specific autoimmune conditions or psychiatric conditions were observed. For psychiatric, they actually were the same across the database. Anxiety, sleep disturbance, and depression were the highest across both databases for vitiligo patients. Autoimmune, atopic dermatitis was one of the top three for both cohorts, with some differences, though, in the second and third most prevalent across the two different databases. And similarly, we did see again, this was a time two analysis. So using a Cox proportional hazard model to then identify the risk of developing any psychiatric or any autoimmune comorbidity over the follow-up was higher for patients in the vitiligo cohort compared to the control cohort, with a similar hazard ratio and also significance value across the published study and what we saw with the replication. And so again, this primary conclusion, controlling for additional factors, we are seeing that hold across the data sets. And again, with this replication, the only change needed to be swapping out the data source that was referenced. So there's known limitations in using, say, closed administrative claims data that may actually impact the generalizability of these findings outside of the replication. For example, we're only really seeing a snapshot of a patient's care during a period of enrollment within a specific health plan. And also many socio-demographic and patient-reported, obviously, factors are not in consideration or are poorly captured in this type of data. So while it's important to replicate findings across databases of the same variety, so ensuring that this study design translates from one EHR data to another, from one administrative claims data set to another, it's also important to see how these findings generalize across different data sources. So kind of linking back to the Bradford-Hill criteria, you want different people under different circumstances, and same with your data, different similar people in terms of population, but they are different people and also much broader enriched study design and variables. can help to support the concept of replication and consistency. So while Catherine will discuss OM1 data, in terms of the multimodal data and its richness in more detail, I'll briefly, prior to that, provide some data on how the case cohort for this study, so these patients with diagnosed vitiligo, which we replicated in IHD using OM1's vitiligo data, confirms these findings again, but also expands the findings from the original study in ways that are unable to be captured using the original data sources. So in the original analysis, on average, what we had was around most patients, their time at risk was close to around 3 years. And the average time for an autoimmune condition to be diagnosed following their initial diagnosis of vitiligo within the data source was around 3 years. And so using the vitiligo cohort within IHD from OM1, we capped the follow-up to a similar 3 years to see how the incidence rates compared. And within our Tesla closed claims data and also the OM1 vitiligo cohort, similar magnitude in terms of the incident rate of any autoimmune comorbidity during follow-up around 10% in Tesla and 11% in OM1. But looking at the data from OM1, which includes registry and EHR data and this multimodal approach to having additional information for patients, gave us a longer follow-up period. So we had closer to 4.5 to 5 years of follow-up for these patients, and seeing that the average time to instant diagnosis within that group is actually closer to 4 years. And in allowing the full follow-up within that data, we saw a higher disease burden that potentially is limited by follow-up in administrative claims data, closer to 15% in the database. So we also, with this longer follow-up, were able to look and compare to other publications and insights that have been recently published among this cohort, including a study in 2023 that used market scan data and only 1 year of follow-up period to identify first-line treatments and identify switching and other treatment patterns over that 12-month period. Within that publication from last year, one of the takeaways from the conclusion was limited switching from that initial treatment over a 1 year period. So in using these defined treatment patterns within the vitiligo data, we're able to also look at the lines of therapy for initiating patients and follow them over a much longer time period, and confirming again that we are also, when you're expanding that beyond 1 year, seeing limited switching after first-line treatment initiation. And finally, it's well known that information such as race and ethnicity are fairly poorly recorded within claims databases. And so that is something as well in the OM1 data, having access to more rich race data and also ethnicity incorporated in there can even improve upon the findings that were initially published. So we had about, as similar to most databases, close to half of patients did not have a race variable coded. And because of that sample drop, they weren't included in the initial Cox proportional hazard model. But in the OM1 data, we're able to do that. And we had similar findings in terms of the other coefficients, but in including this new more populated race data, we're able to see a statistically significant increased risk of incident autoimmune conditions among American Indian or Alaskan patients compared to white patients, and it being statistically significant, an interesting finding that was not captured within the original data set. So in that sense, this concept of replication is important, and then in addition to that, maintaining your study design and facilitating that with a different data set is something that's needed to confirm the findings that is initially published, but additional data sources in addition to that conclusion can be expanded on with rich data. And the concept of replication isn't limited to just the initial type of data. It can be expanded and brought on to different data sources. And so with that, conclude my section and pass it off to Catherine from OM1. And in the meantime, as we are switching, we do have an additional poll that is available on the screen.
So give everyone a minute to do that. But in the meantime, thanks, Mike. So I'm going to talk a little bit about a little deeper dive into the data and what can be done. It was very interesting to see how in this exercise, going from comparing two claims data sets in terms of replication and reproducibility, moving on to the EMR and claims and additional data sets to see how we could do similar studies. But what it brings up as well is, it's limiting in terms of all the things that we can now do from a clinical point of view. So one of the things that we're definitely thinking about is not only reproducibility, but how do we bolster what we're doing, and it's to really better understand what's happening in these areas, because obviously reproducibility and to some extent replication is really trying to strengthen the evidence and to strengthen the value that the data can bring. And what we're thinking through is how do we pursue that in terms of treatment effectiveness and other outcomes in chronic autoimmune diseases, really targeting as we move towards more personalized treatment choices. How do we bring that together with the EMR and the ability to do things in something like IHD, where we can reproduce the data and the evidence based on a similar study design, really strengthens what we're trying to do. Part of the reason we chose to concentrate on vitiligo and alopecia today is that they're both conditions we're only really recently seeing novel therapies emerge, and there's a lot to learn still about which patients are most likely to benefit from which therapies and when. For vitiligo, topical steroids and calcineurin inhibitors and ultraviolet phototherapy are still the basis of treatment. But as often is the case with autoimmune diseases, being on long-term steroids is really not a good option. And it's just not a long-term solution. As Beg mentioned earlier, there are systemic therapies currently in late-stage trials for vitiligo, and some that were approved within the last couple of years for alopecia. So they're certainly interested in what real-world data can tell us about disease heterogeneity. So some of the things that we saw in terms of comorbid conditions in the analysis that Mike did, about current treatment pathways, and ultimately about the unmet need in these conditions. So despite some of these recent advances for both vitiligo and alopecia, there really remains a challenge to manage. So we see differences in response based on bodily location, and remains restrictions based on extent of disease and age. For example, in clinical trials, topical ruxolitinib showed the best results on the face in vitiligo, which is important, but lesions on the hands and feet and some other areas showed the least improvement. So there were definitely differences based on location. And ultimately, it is restricted to use on 10% of body surface area. So it's not a one size fits all necessarily. And the other thing to remember is that location really does matter. So in terms of achieving treatment goals, we've seen this more extensively studied in conditions like plaque psoriasis, and some of the most challenging to treat cases are based on their location. And location also can play a big role in the patient's perception and their experience of the disease and of the treatment. So here we see some excerpts from some actual patient notes, and I show them just so everyone can get a sense of the richness of the data, but also the complexity from the point of view of teasing out treatment patterns. So, we can see that there's some complexity in terms of instructions to the patient. So using multiple topicals and starting with one, switching to another after a certain amount of time. We often see the dermatologists providing qualitative assessment of the condition's severity. and as well as the extent and location of disease. So these are all important aspects as we try to really understand what works for which patients, how well it works, and for how long. And in addition, really how those treatment decisions are being made and what's influencing that. And the only really way to process all of this data, because if you think about tens of thousands or sometimes hundreds of thousands of patients, and certainly many more visits associated with that and notes, right now for consistency, you really have to automate it. And that's, an area where we're using AI in a variety of forms to do that with a clinical eye also, to make sure that we're really getting the context of what's happening. And then this is another way of bringing in some standardization about how some of this unstructured data, which can be unwieldy, can really be brought into structured data and ready to be analyzed. So here we have a snapshot of the vitiligo data in OM1. So this is based on dermatologist-treated patients. So Mike showed you earlier some of the work he did on IHD, and specifically looking at the development of comorbid autoimmune disease and potential differences according to race and other demographics. And here you see an age, and this is a snapshot of the prevalent population, so not age at onset, but you will see that you see the bimodal distribution, which is what we'd expect to see with the disease, with some patients with an early onset, typically getting diagnosed around 10 years old or so, and then the late onset or late diagnosis patients that you see later on. Male-female ratios is expected, about 1-to-1. And again, we still see, the dominant treatments being corticosteroids and the calcineurin inhibitors, but we are seeing a steady increase in topical Rux use. And now we're seeing it in over 11% of the cohort, and that's changing over time. So this starts to give you a picture of the distribution of the patients and sort of what we can do in terms of thinking about different phenotypes. So is early onset different than late onset? Are they managed differently? Are the treatment options different based on age? Is severity different? So there's a lot of ways that we can think about incorporating these data. So it's important to note, as we, part of this was really thinking about comorbid diseases and the analysis that was done to show consistency of, why this is important. But, 1 in 4 patients with an autoimmune disease are going to be diagnosed with at least one other autoimmune disease. And it may be even higher given what we know about underdiagnosis. In more rare cases, patients can have what's referred to as multi-autoimmune syndrome, so 3 or more conditions to manage simultaneously. And these combinations ultimately can present complicated treatment decisions, and care is not always well coordinated across specialties. And circling back to that replication of comorbid condition, if we look here, and this is looking at comorbidities, not necessarily incident. So timing of diagnosis, they could have been, for instance, diagnosed with rheumatoid arthritis prior to the diagnosis of vitiligo. But you see there's almost 4% overlap with alopecia areata, but also patients with comorbid rheumatologic and gastro conditions that might be important in respect to treatment decisions, particularly as systemic options emerge, and some of them overlap in terms of effectiveness across more than one condition. So depending on what the dominant disease in terms of severity is, there may be some trade-offs about what could be used amongst these patients. And one thing this can show also, there's an opportunity to be better at detecting potential comorbid autoimmune diseases in these patients, and to potentially speed time to diagnosis if we can identify them in these data sets of patients at risk of being or likely having a phenotype that might be associated with one of these other conditions. And here, just a quick peek at the alopecia areata patient population. Definitely a different age distribution, a higher proportion of females compared to males. And then also there's definitely different phenotypes within the data set in terms of totalis and universalis versus the more patchy disease. And finally, just a few thoughts to circle back where Meg started when talking about the condition. So, another aspect of context, which is part of the criteria in addition to replication, is having to incorporate, if we're really going to understand the patient journey, more of an interpretation of the evidence we do generate. So last year at ISPOR, we presented some data both on vitiligo and alopecia areata, looking both at social determinants of health and also psychological comorbidity in vitiligo and alopecia, because both of these diseases are disfiguring. They can come with a heavy psychological burden. And as we look to improve outcomes for these patients, based on solid and contextually reproducible evidence, we have to also do it with an eye towards equity and access to effective care and understanding the outcomes that are meaningful for these patients. So by having access to data that both touches on the clinical, but also risk factors and drivers of potential effectiveness that may not be evident just from diagnosis codes, for example, is really important if we're going to provide actionable evidence in the field for comparative effectiveness.
Thanks, Catherine. So in closing, we've talked not only about replication and the Bradford Hill criteria, but we've touched on the idea of expansion as well. Thank you, Mike and Catherine, for highlighting how important and helpful it is to have expanded data. At Panalgo, we think about data and evidence in much the same way that Georges Seurat thought about his paintings. I never realized this until recently, but in talking about his dots of paint or pointillism technique, he said, great things are done by a series of small things brought together. And we agree. Great things can be accomplished, as you've seen, by combining multiple sources of data, looking at multiple cohorts, attempting to replicate on your research and expand your research with a deeper look into the patient journey. And great things come from partnering in the generation of evidence for those data. Great things are going to come of our partnership, Panalgo's partnership with OM1. So thank you so much. Oh, wait, before I say thank you, let me just tell you how the poll questions are looking. So back to the list of celebrities who may or may not have suffered from vitiligo. The correct answer is that everyone except for Zach Galifianakis is a vitiligan. There was a story in the news recently about John Hamm, the Mad Men actor, developing vitiligo because of the stress of the pilot episode of that television series. And Catherine's question about underlying the percentage of overlap between vitiligans and alopecians, the correct answer, which was the majority answer, is any of those percentages depending upon the underlying population. So I want to thank you for joining us today, and I want to say that we're happy to take your questions or have you share your insights. Let's welcome Mike and Catherine back to the screen, and we'll open the chat channel to see if any questions come in.
Yes, thanks, Meg. And thank you all for the presentation. It was really great to hear from you and very interesting. For our audience, as Meg said, you can submit your questions in the Q&A tab, and we'll try to get to as many as we can in the time we have left. So let's go ahead and ask the first question. Does IHG use a common data mode model?
Do you want to take that, Mike?
Sure. Yeah, so that's one of the key aspects of the platform that makes particularly replication possible. So data sources can be mapped to the common data model, and there are some, obviously, as we discussed, some nuances between data sources based on the types, like administrative claims data. You'll have continuous enrollment that you won't have in EHR data. But overall, the concept of looking at the data and identifying encounters, and within those encounters, understanding all the information from a patient, that data model is translatable, and what makes the ability to replicate a study within the platform independent of the data source that's being used. So, in addition to the speed and you don't have to rely on coding. I mean, that's a big piece of it. But the common data model is what mostly speaks to the replicability aspect of IHD.
Thanks, Mike.
Yeah.
Okay, wonderful. We have another question here. Are the various OM1 data sets all structured the same?
Catherine?
Sure. So our premium data sets, which are mostly around chronic conditions, are structured the same in terms of the table structure. Some of the differences are in some of the variables available, but they're typically under the same observations and tables across the structure. So yes, it is a similar structure across all of them.
Catherine, I have a question for you. How many conditions does OM1's do OM1's data sets cover? Is it like, I think you have a couple dozen now.
Yeah, so it's about 26. So that's in terms of the data sets that are that we have fully curated across rheumatology and gastro and derm and cardio. But in those therapeutic areas, especially the ones that we concentrate in, we often do custom cohorts of some more rare conditions, sometimes things where we can curate them on demand. So we're always expanding a little bit.
Yep, and we have the potential to map and load all of them into our platform because we've already developed the sort of common data structure to host all of OM1's data set offerings. So that's part of the brilliance of our partnership.
Oh, wonderful. I love that added commentary. Thanks, Meg.
Sure.
Let's go to our next question here. If I wanted to explore OM1's data in IHD, how do I make that happen? And how long will it take me to get access to the data?
Well, that's a great question. Do you want me to start, Mike, and then do you want to chime in?
Yeah, I think that makes sense for you.
Yeah. So if a client already has access to IHD and they would like us to, so to speak, turn on access to a certain OM1 condition-specific data set, we can do that within a matter of 5 to 10 days. That's the beauty of data on demand is because the data are there, and we've ironed out this agreement with OM1, between OM1 and OM1 and Panalgo, to make those data available. We can do that pretty quickly. If a client is interested in using OM1 data in our platform, but they don't yet have a relationship with Panalgo or don't yet license the platform for use, we can draw up a number of scenarios where we bundle the data, the platform, and analyst support in whatever's the most fit for purpose way for the client to interrogate those data. It might take a little bit longer to work all that out, but we've got lots of options, and you can sort of choose your adventure and we'll work with you to make sure that whatever we present to you fits your research needs as closely as possible. Anything to add, Mike?
No, I think you covered it all there. Nothing to add on my end.
Okay, thanks, Meg, for the breakdown. Let's see. Are autoimmune conditions like vitiligo and alopecia areata becoming more common?
I'll start, Catherine, but then I want you to chime in. I read this stunning, when I was prepping for this webinar, I read this sort of stunning figure from, I think, the NIH, that autoimmune diseases as a collection of conditions are increasing, I think it's like 3 to 12% annually across the globe. And it's gotten to the point where 1 in 5 of us is likely to develop some sort of autoimmune condition in our lifetime. And the reasons for that, you could probably guess, include climate change, environmental stressors, upticks in obesity and cardiometabolic diseases, and COVID-19, things that carry sort of inflammatory processes with them. And that all can lead to, a much higher prevalence of AI diseases. And as you said, Catherine, treating them, if you're a person with multiple AI conditions, then treatment decisions can get very tricky. Is that right?
Yeah, the other thing I would add, because I saw something recently as well where it was interviewing patients, but it did a lot of self-reporting of patients who had vitiligo but were not being actively managed clinically. Either there are milder cases or there weren't opportunities to get treated, but it'll be interesting to see also the effect of there being more effective treatments being developed, more systemic treatments, whether we see, not in addition to possibly being more cases, but actually more presentations clinically as the treatment options improve.
Yeah. Yeah.
Okay. Thank you so much. Any last words from either three of you that you'd like to say that maybe you didn't have time to mention, or you thought of a question like Meg on the spot?
Actually, Catherine, if I may, I have another question for you, because
Sure.
I love your rich data source. When I was just looking up the background on alopecia, I was really struck by how much of the treatment decisions is based on age quite a bit. It seems like with little kids, there's a kind of a wait and see, that's tried first, and then also like the location and the extent and the duration. And do you see that too in the alopecia data, that it really depends on a lot of factors? And do kids who have alopecia, do they recover? Does that happen?
Yeah, there is some variability. So particularly with the most common, the patchy form of alopecia, there's a significant proportion of it that spontaneously gets better. So, but then it's also at risk of relapse, and it's stressful thinking, oh, it's going to come back, if there's an environmental stressor or something like that. So I think when we do see a lot of variability with children, I think there's a reticence to do anything too strong, certainly in terms of chronic steroid exposure and that kind of thing. So there is differences age-wise, but we see a lot of variability, and also sort of willingness or wanting to do anything too strong if there's a potential for it to regress.
Yeah. I had a colleague years ago, she's still one of my favorite colleagues to this day, whose daughter had idiopathic alopecia. They tested her for all kinds of things, but in a gesture of solidarity, her mother shaved her head so that they could look alike, which I thought was a really neat thing for a parent to do. So,
Especially
oh,
for a child, I think that's nice to show the support because, as a kid, everything is kind of a big deal.
oh yeah. I think her daughter was 5 or 6 at the time, like, so just entering elementary school. And yeah, I thought that was neat. Hey, Sydney, I see another good question for Catherine.
Yeah, I see it here as well. So, what PROs are available in OM1 alopecia and vitiligo data?
So in routine practice, we don't see a lot of PROs being done. So, what does come through the EMR, we do pull through. What we do instead, to augment what we have, is to go into our network and actually administer PROs to bring back into the data sets. And that's an ongoing effort that we're doing right now, because there really isn't a lot of PROs in the space.
Okay, fantastic. Thanks for that last question. I think we've reached basically time. So I want to say thank you so much to our 3 expert panelists for your time today. It was a pleasure to have you here. I hope you enjoyed yourselves, and it was really great to hear all of the data that you presented.
Thank you, Sydney. Thank you, Mike. Thank you, Catherine.
Thank you.
Thank you.
Good afternoons, evenings, all.
In closing, we hope you enjoyed this HealthEconomics.Com webinar sponsored by Panalgo, and I hope you enjoy the rest of your day.